time now to open the junior researchers session. So for people in the room, I changed my job. No, it's because Dr. Philippe Blanche-Maison cannot come, so it's my job now, but I'm not vascular physician. Explain to you. So first searcher, it's Céline PISIBON from Nice, who speak from melanocyte differentiation. It's to you.
So good afternoon, everyone. I'm Céline PISIBON. First, before I start, I would like to thank the organizing and scientific committee for giving me the opportunity to present here today. I'm from the team of Corinne Bertolotto and Robert Balotti, and today I'm going to talk about alteration of melanocyte differentiation process at the crossroad of skin pigmentation disorder and melanoma resistance. So melanocytes are specialized cells that you can find here in the epidermis that produce the pigment melanin. And pigmentation is a complex process that begins with the synthesis of melanin within the melanosomes, here that you can find in the melanocytes, that will be transferred to the keratinocytes in order to protect the skin from the UV. But what are the mechanisms that are involved in skin pigmentation regulation? So when you expose your skin to UV light, UV radiation will induce DNA damage in the keratinocytes that will activate p53, upregulate alpha MSH, which is the melanocyte stimulating hormone that will be secreted by keratinocyte and will bind to the receptor in the melanocytes, the MC1R, and this binding will lead to activation of MITF, which is a major transcription factor that will increase the expression of pigment genes such as tyrosinase, TERP1, and DCT. And this will lead to production of melanin, melanocyte formation, and that will be transferred to the keratinocytes in order to protect the skin from UV. So here you can see that MITF is a master gene of melanocyte differentiation. It has a key role in skin pigmentation, but also in different diseases such as cutaneous melanoma. But more than 650 genes have been involved in pigmentation. In fact, what I just showed is for skin tanning, but also in congenital and acquired pigmentary pathologies such as lentigo and melasma, and other severe diseases such as cutaneous melanoma that I will talk about today. So cutaneous melanoma is a cancer that derives from melanocytes. It represents less than 10% of skin cancer. In fact, in 2022, more than 300,000 new cases worldwide were diagnosed. And this cancer is responsible of 80% of mortality that is associated to cutaneous cancer. So what about treatments? For cutaneous melanoma that is still localized, the treatment relies on surgery, but it's a cancer that evolves very rapidly. And for these patients, for metastatic cutaneous melanoma patients, the treatment relies on targeted therapies and/or immunotherapies. But what you have to keep in mind is that 50% of patients are resistant or develop resistances to these treatments. But what are the causes of resistance to targeted therapies? So it can be explained by genetic modifications, but today we will only focus on non-genetic modifications that are reversible and that can be caused by the tumor microenvironment, the treatment itself, that will induce what we call a phenotype switch or pseudo-EMT. And in a very simplified way, here on the left, you have a differentiated phenotype with cells that are more sensitive to therapies, characterized by the expression of MITF, that can switch through epigenetic changes to a more dedifferentiated mesenchymal phenotype with cells that are resistant to therapies and cells that are characterized by the loss of the expression of MITF and the gain of the expression of some markers such as AXL. But how can we prevent or revert resistances? And also how to identify epigenetic players that revert the differentiated phenotype. So I don't know if you're familiar with this epigenetic players notion, but what you have to know is that the chromatin can be in two different states. So in a closed chromatin, a repressive state where there is no transcription, and an open chromatin, which is an active state with transcription that is allowed. And these states can be modulated by some epigenetic players, such as histone modifiers, like histone methyltransferases, histone deacetylases, that will add some acetyl groups or methyl groups that will change the condensation of the chromatin. And this is important because, as we saw, in the dedifferentiated phenotype, we have cells that have lost the expression of MITF that could be due to a condensed chromatin within the promoter of MITF. So the hypothesis was that epigenetic drugs that restore MITF expression and differentiation will also restore sensitivity to target therapies. And to answer this hypothesis, we use an epigenetic drug library in dedifferentiated cells. So how we did, we transfected our cells with a plasmid containing a tyrosinase promoter that is linked to a luciferase reporter. And the hypothesis is that epigenetic drugs that target these epigenetic players will increase the expression of MITF, activate the tyrosinase promoter, induce the transcription of luciferase, and we will be able to measure it by light emission. So here are the results that we obtained. And what you can see here on this plot, UNC 0642 and BX12094 are two inhibitors of the methyltransferase G9A, or EHMT2, and that are in red and pink, and they should increase MITF expression, but because we saw an increase of the light emission. So to validate these results, we looked at MITF expression at protein levels, and as you can see, with a dose response of these two inhibitors, we have an increased expression of MITF. And to go even further, we went with some siRNA transfection in order to decrease the expression of this methyltransferase that catalyzes the H3K9Me2 modification that is associated to transcription repression. And upon the knockdown of G9A, we have an increased expression of MITF. And if we go back to the hypothesis, it was that higher expression of MITF and differentiation should increase the sensitivity of these cells to targeted therapies. So that's what we did. We tried to use the targeted therapy inhibitor and also the UNC 0642, the inhibitor of G9A. And we combined these drugs in order to see if we could resensitize the cells to target therapies. And that's the result that we got. We decreased the number of cells at the end of 72 hours of treatment. So G9A inhibition induces redifferentiation, but also resensitizes resistant melanoma cells to BRAF inhibitor. And one mechanisms that lead to this transcription repression induced by G9A is through a protein called CTBP1 and 2. So CTBP1 and 2 form a family of transcriptional co-repressors. They are implicated in EMT, in proliferation, metastasis, resistance to treatments in different cancers, and they have a role of negative transcription regulator. So how they work, CTBP1 and 2 that are here can interact with some epigenetic players, histone modifiers, such as histone methyltransferases, as we saw before, G9A, and this form a co-repressant complex that, associated to a repressor, will induce chromatin condensation and transcriptional repression. So why working on CTBP1 and 2? Because first, they favor this mesenchymal phenotype, and also because inhibition of CTBP1 and 2 was reported to favor MITF expression and melanocytes differentiation. So the main question of my PhD was, are the CTBP's co-repressors implicated in melanoma plasticity and resistance acquisition to therapies? So to answer this question, we used an experimental model of BRAF V600E human melanoma cells. So the BRAF mutation is the most frequent one found in patients harboring melanoma. And we use sensitive and resistant cells to target therapy. And first, we validated the phenotype of these cells with a dose response of targeted therapies. And you can see that the sensitive cells are really sensitive to this drug, and the resistant are really resistant to targeted therapies. And to go even further, we characterize these cells, and as you can see at protein levels, the resistant cells have lost the expression of MITF, have gained the expression of AXL, and we also looked at the expression of CTBP1 and 2 that do not seem to be impacted by the sensitivity or the resistance of the cells to targeted therapies. So using this experimental model, we use a loss-of-function approach using inhibitors and shRNA in order to decrease the expression of CTBP1 and 2. And we will start with the inhibitor part. So we use the NAC 95397, which is an inhibitor of CTBP1 and 2 that will focus on the complex, and this inhibitor will disrupt the complex, so we will lose the interactions with the partners. So first, we did a dose response of this inhibitor in order to find a concentration that do not affect the proliferation itself, as you can see here in the crystal violet. But what is interesting is when you combine this drug to target therapies, we decrease the number of cells at the end of the treatment. And this result has also been confirmed by some cell countings, where you can see a decrease of 50% when we combine target therapies with the inhibition of CTBP1 and 2. So pharmacological inhibition of CTBP1 and 2 resensitizes resistant melanoma cells to target therapies. And then in the second time, we looked at shRNA in order to decrease the expression of CTBP1 and 2 and see if we could confirm the results that we got with the inhibitor part. So that's what we did. We infected our resistant melanoma cells with shRNA in order to decrease the expression of CTBP1, CTBP2, and we also included a condition where we knock down CTBP1 and 2 together. And then we looked at the effect of this knockdown on cell proliferation first. And as you can see, knockdown of CTBP1, CTBP2, and CTBP1 and 2 together do not affect the proliferation itself. But what is interesting is when you combine this knockdown with PLX, you decrease the number of cells at the end of the treatment. And this result has also been confirmed with the two different resistant cells that we used. So these were results that were done in vitro, but we also went with some in vivo experiments using immunodeficient mice, NSG mice, where we subcutaneously injected these mice with resistant melanoma cells, knocked down or not for CTBP1 and 2, and these mice were treated with targeted therapies. And what you can see is that the group of mice treated with targeted therapies and that harbor the tumor knockdown for CTBP1 and 2 present a reduction of the tumor volume in comparison of the other groups. So knockdown of CTBP1 and 2 resensitizes in vitro, but also in vivo melanoma cells to target therapies. But now the question is, what are the molecular mechanisms leading to this resensitization? And to answer this question, we perform RNA sequencing in order to compare SH control versus SH CTBP1 and 2, and we obtain different results. So first, we saw that knockdown cells for CTBP1 and 2 present an enrichment of melanocytic markers, so differentiated markers, such as MITF, and we confirmed these results at protein levels by Western blot, where you can see that the knockdown of CTBP1 and 2 in knockdown CTBP1 and 2 melanoma cells, we have an increased expression of MITF, also associated to a decreased expression of AXL, the marker of resistance. So inhibition of CTBP1 and 2 redirects melanoma cells towards a more melanocytic, differentiated phenotype. And the second thing that we saw was that in deficient CTBP1 and 2 melanoma cells, we have an inhibition of the UPR response. And UPR stands for unfolded protein response, which is a cellular stress response related to the endoplasmic reticulum or ER stress. And to validate these results, we looked at one major effector of this ER stress called ATF4. That we can see here in Western blot, we validated the knockdown of ATF4 when we knocked down CTBP1 and 2. And then we looked at one of the targets of ATF4, which is SLC7A11, that we also confirmed at protein levels, where you can see a decrease of the expression of SLC7A11 when we decrease the expression of CTBP1 and 2. So attenuation of ER stress response by CTBP1 and 2 inhibition contributes to the resensitization to targeted therapies. So SLC7A11 is a cysteine glutamate antipoda that will allow the entry of cysteine and export the glutamate, and it's an antipoda that is essential for detoxification of cells. But how SLC7A11 expression is regulated? So we saw that ATF4 can regulate the expression of SLC7A11, and that's what we have. But could it be also an epigenetic regulation? And to answer this question, we perform attack sequencing, which is a technique that will allow us to see where the chromatin is open or not. And we perform this sequencing on sensitive and resistant cells. And you can see that we have an upregulation of this peak in resistant cells just above the promoter of SLC7A11, confirming the results that we have that resistant cells express higher expression of SLC7A11 compared to sensitive cells. And then we also perform some ChIP-seq in order to look at the mark H3K9 acetyl, which is an active mark that will allow the transcription of the genes. And what you can see is that in knockdown cells for CTBP1 and 2, we have a decrease of this peak just above the promoter of SLC7A11, also confirming the results that we have as decreased expression of SLC7A11 while we knock down CTBP1 and 2. But SLC7A11, is it implicated in melanoma resistance to targeted therapies? So to answer this question, we use two inhibitors that are known in the literature called Erastine and sorafenib, and we combine these inhibitors with targeted therapies, and as you can see, in the differentiated cells, when we combine the two treatments, we decrease the number of cells at the end of 72 hours of treatment. So resensitization of melanoma cells seem to be mediated by SLC7A11 antipoda. So to conclude, we saw that differentiated phenotype with cells that are sensitive to therapies can switch to a more dedifferentiated phenotype with cells that are resistant to therapies. And if we look closer at this epigenetic landscape and we target these epigenetic players such as GNAN-A or CTBP1/2 by pharmacological inhibitions or by shRNA in order to decrease the expression of them, we go back to a more differentiated phenotype with cells that are more sensitive to therapies and also characterized by increased expression of MITF and decreased expression of mesenchymal markers such as AXL. And this resensitization goes through a decrease of the ER stress response through inhibition of SLC7A11 expression. So targeting CTBP1 and 2 or the targets could be an interesting therapeutic strategy to overcome melanoma resistance to targeted therapies. But if we go further, this was in a cancer context, but if we go further and maybe we could apply these strategies and we could extend them to non-malignant diseases such as pigmentation disorders like hypo or hyperpigmentation. And with that, I would like to thank all the members of my team and thank you all for your attention.
Thank you, Céline. Do you have any questions for Céline? Marek?
Do you have an idea whether reconditioning of these melanoma cells, resistant melanoma cells, with your epigenetic approaches, does influence metastatic potential of these cells? Have you ever looked into this, or is it the future direction?
Yeah, exactly. That was a part of my PhD project where because we thought that epigenetic regulators could influence resistance to therapies, but also the plasticity in general. So we tried to see if the inhibitors or the knockdown cells using shRNA, for example, could modulate the migration of the cells. So we did some Boyden chamber assay, invasion assay, but unfortunately we did not see any changes.
Good or better direction?
Thank you for the nice talk. I have a question, like this mechanism that you described through the CTBP1 and 2 transcriptional co-repressors, is this specific for melanoma or melanocytes, or is this a mechanism that you have in all cells? Because in that case, if you would targeted, would you expect some effects on other cells?
That's a really nice question. To be honest, TBP1 and 2 has been implicated in other types of cancer, and this EMT, so phenotype switching, is occurring in a lot of cancers. So I'm guessing that what we have found could also be implicated in other types of cancer, and we will maybe try to do that.
Thank you.
Thank you.
Lionel?
Yes, thank you for your call. Inside your abstract, you mentioned pharmacological inhibition of the. So, which kind of drugs could be efficient of this field?
Sorry,
Which kind of drugs could be efficient of this field? Because so you mentioned that could be an option for pharmacologists. Like
yes.
me, for example, you know. And so, how could you imagine something that could be relevant to control by, I don't know, chemical or biotech, I don't know, any kind of things that could be regulated? Did you test something or not yet? So.
Yeah, that's a really nice question. It's true that nowadays epigenetic drugs are more and more often on the market, and they can be implicated and advanced like the treatments for cancer patients. In our cases, unfortunately, the drug that targets CTBP1 and 2 could not be used in vivo. We tried it with some mice experiments, but there are new drugs that are found. In fact, there's a paper of Filograna in 2024 that has shown that a specific CTBP1 inhibitor could be used in NSG mice, and we should test it. So this could be one part. And then with the epigenetic drug that we have conducted, the drug assay that we have conducted, we have chosen drugs that were already in clinical trials. So I'm guessing that if we could go further, it could be interesting for melanoma patients.
I had a quick question for you. So treatment with the epigenetic modifiers sensitizes your melanoma cells to BRAF inhibitors, right? And then you have done this assay in not SCID mice. So what have you thought of doing it, or have you done it in immunocompetent mice? For example, from the Yale University, they have this Yammer cell line, so that way you can see if there is any response in terms of the immune cells.
Yes, for sure. That's what we want to do next. So it's true that we use only human melanoma cells, so that's why we went with immunodeficient mice. But one part of my project was to also work on murine models, so UM and B16 mice, for example. And also, I didn't have time to show that today, but we are also interested in the immunotherapy part, because we also know that cells are resistant to immunotherapies, and we have conducted some experiments regarding CTBP1 and 2 knockdown cells, if they can influence immune response. And I just have some few results, so I don't know yet very well, but it could influence probably T-cell activation and NK cells. So it could be interesting also to look at the effect on other models and immunocompetent mice, for sure.
Thank you, Céline.
Thank you.
We don't have time for other questions.